In this tutorial we will discuss the differential gene expression module. The objective of this module is to compare the expression of genes between two determined groups of cells. Beforehand, the project has been loaded and the modules HTO demultiplexing, quality control, gene selection, principal component analysis, t-SNE, and UMAP analysis, clustering and marker genes have been applied. To perform differential gene expression analysis, we need to define two groups of cells. This module allows you to define cell groups very precisely from the previously calculated cell annotations. First, for the first group, we must choose an annotation, that is, define the notation associated with the cells that we will use to determine the group. For example, we can choose the identity provided by the HTO demultiplexing, that is, the original sample of cells. We can also choose one of the clustering analyses we have performed previously, for example here, the one based on the Leiden algorithm. We then see the cluster numbers appear. Here we can choose clusters 1 and 5 by deleting the other cluster numbers. For cluster 2, we will choose in the same way clusters from the Leiden algorithm. And we will choose for example cluster 4. Finally, we must choose in which representation to visualize the chosen groups of cells. We can choose the PCA representation or the dimensional reductions t-SNE or UMAP. Here we will choose UMAP. We can then click on Next. We have several pieces of information. On the right we have the representation we have chosen, here UMAP. In color we see the groups of cells that we have determined, Group 1, which is cluster 1 and 5, Group 2 which is cluster 4, and in blue, the cells that have not been selected in one of the two groups. Here we have the indication of the annotations associated with the groups, 1 and 5 for Group 1, and 4 for Group 2. As well as the number of cells that are associated with each of the two groups. If the groups you define overlap, that is, there is an intersection of cells between the two groups, ShIVA warns you by indicating the size of the intersection and suggests that you do not perform this analysis, but leaves you free to do so. It is not advisable to perform a differential gene expression analysis between two groups with a non-zero intersection. Once the groups have been properly defined, we can apply the differential expression analysis. If you have followed the tutorial in the clustering and marker genes module, you will recognize this box which requires the same parameters. First, we need to set the minimum fraction of cells expressing the gene. Here we have for example a value of 0.25. With this value, the analysis will only consider genes that are expressed in at least 25% of the cells in the cluster. All genes that are expressed in less than 25% of the cells will be ignored. Next, we need to determine the minimum log fold change. The log fold change is the log ratio of the average expression in the cluster to the average expression outside the cluster. The analysis will only report genes whose log fold change is greater than the value indicated. Here we will consider those with a minimum log fold change of 0.5. We must then choose the method used to calculate these marker genes. There are several methods available. By default, the Wilcox method, that is, the Wilcoxon test, is applied. But you can choose another method. The last thing we must choose is whether we want to display the positive markers, or all the markers. The positive markers are the genes that are overexpressed in the Group 1 compared to the Group 2. Negative markers are genes that are underexpressed in the Group 1 compared to the Group 2. Here, it should be noted that the positive markers for Group 1 are the negative markers for Group 2, and conversely the negative markers for Group 1 are the positive markers for Group 2. We will deselect this option to have all the marker genes and run the analysis. Once the result is obtained, several pieces of information are at our disposal. In the first tab, we have a summary of the analysis that has been carried out with a reminder of the groups and the parameters chosen. The table contains the first 200 genes identified, but the complete result can be downloaded via the download button. This table contains the name of the gene, the adjusted p-value, the initial p-value, the average log fold change, the percentage expression of the gene in group 1, the percentage expression of the gene in group 2, and the difference of the percentages. We note here that log fold changes can be negative, indicating genes that are more expressed in group 2 than in group 1. In the second tab, we have at our disposal representations in the UMAP of the expression of the 9 most significant marker genes to identify whether they are indeed markers of the groups of cells we have chosen. The other 4 tabs give access to functional enrichment analyses. These analyses take the identified genes as input and look to see if these genes are enriched in one of the predetermined annotation modules. We have 4 types of annotation modules. The first 3 are from the gene ontology annotations. These are biological processes, molecular functions, and cellular components. The 4th type concerns the annotations provided by the KEGG database. When you click on one of these tabs, the analysis is launched, because the analyses are not launched a priori to save computation time. For example, we choose the biological processes by clicking on the tab. The analysis is launched. The result that is provided gives us the annotations that are significantly enriched in the genes identified by the differential expression analysis. Only the first annotations are shown in this graphical representation, but the full set of results can be downloaded via the download button. In this graphical representation, the lines are the annotations, here, biological processes. The x-axis represents the percentage of genes of this annotation, which are in the list of differentially expressed genes. The color of the bubble represents the intensity of statistical significance. The redder the color, the greater the statistical significance. The size of the bubble represents the number of differentially expressed genes associated with that annotation. You can proceed with the other analyses if they are of interest to your scientific questions. Once you have completed these analyses, you can start again with other groups of cells to deepen your understanding of your dataset. You can also move on to the interactive exploration module, which is the subject of another tutorial video.